Denise O'Meara is coming up next. Denise is a lecturer in WIT. She's one of our most eminent geneticists, and thankfully she's specialised in protected species and rare species. She's done a lot of work with us on bats, but also looked extensively at carnivores, including pine martens. So she's going to give us some insight into her genetic works on pine martens. Thanks, Denise.
Hi, thanks very much for having me. I'm sorry I'm not there in person today. I got COVID last week and I decided I didn't want to be responsible for rebranding the Pine Marten Symposium, the COVID Symposium. So I've spared you all that. Thanks very much. I'm looking forward to having a chat to you about the genetics of the Pine Marten in Ireland today. Move on my own slides. Okay, so the paper that I'm talking to you about was actually published last year. We published it in the Biological Journal of the Linnean Society, and it involved a big group of people. So Catherine O'Reilly was the lead author on the paper, and Catherine did all of the molecular work over a good few years, and herself and Pete would have collected a lot of the samples within kind of the southeast of the country. But then Declan O'Mahony had been involved from the previous national surveys, and then Josh and Dave contributed samples from Northern Ireland, particularly part of Josh's PhD work. And then we have people like Chris Small, who would have independently collected a large number of samples. And we had Kate McInerney from the Vincent Wildlife Trust, and then Ciara Powell, who's now a member of the National Parks and Wildlife. She would have collected some of the samples, as did John Power, who was also a PhD student in WIT at the time. So I suppose my role in this was analysing the data that had been collected over quite a long period of time, and trying to understand it and to maybe make some suggestions as to how we can go and continue to manage the pine marten population into the future, while understanding, I suppose, maybe some of the difficulties that they've overcome historically, which Dave has spoken to you about, and how that has actually imprinted on their genetics today as well. So thankfully, I don't have to go into any great detail about pine marten history in Ireland, other than to say, I suppose, that when we know that pine marten were first discovered to be in Ireland about 2,800 years ago, that was a time kind of in the late Bronze Age when woodland cover was very high in Ireland. And that was kind of, I suppose, maybe the peak of Ireland's woodland cover. And since then, it's been in a state of decline. And there was a very good podcast on last weekend on Lyric FM. It was presented by Anya Murray, and it goes into a lot of detail about the history of woodland in Ireland and some of the political and, I suppose, cultural reasons as to why woodland declined so dramatically in Ireland, and what were the drivers behind that. And I think listening to kind of some of that kind of context helps us to kind of get a better understanding as to why woodlands in Ireland today are, I suppose, you know, at the level that they are. We still have one of the lowest woodland covers within Europe at 11%, and only about 2% or 2.5% of that is actually native woodland. So I think it's worth considering where we've come from to where we are today. As Dave was saying, in the 1970s, you know, the pine marten population was certainly mostly restricted to kind of the west and the Midwest part of Ireland. There were some pockets of pine marten in other locations around the country, but generally speaking, the population was fairly well retracted, and that's, you know, assuming that we had low population numbers historically as well, when the woodland cover was as low as 1% at times historically. So we have a fairly good understanding that pine marten numbers have fluctuated considerably over the last few hundred years, And I think it would be quite safe to assume that the population has expanded and retracted a number of times over that period. But we're fortunate that today that the pine marten population is quite common, and it's considered fairly widespread across the country. But what we wanted to do in this study was to look at the genetic diversity of the pine marten population as it is today, and to maybe get an understanding of how some of these historical retractions shaped the contemporary diversity that we see in the population, and to then get an understanding of what kind of challenges the population might face as it continues to expand and recover across the country. So the map I have here is a map of all of the samples that we use in the study. We've actually got, we had almost 250 samples, and they represent, quite a broad distribution of pine marten within the country. So I think it's a good representation of where things are today. So what we did was we looked at two different genetic markers. We looked at the first marker, which we call the nuclear DNA, or the microsatellite markers. And they're what we use to provide kind of an insight into kind of contemporary genetic diversity. On the right-hand side there, I just have an example of these microsatellites, which they're effectively a repeat region of DNA, and the number of repeats varies across individuals. And it allows us to go on and to statistically analyze the data to see how variable or not variable a population might be. And then the other part of the DNA that we looked at was the mitochondrial DNA. And the mitochondrial DNA is a section of DNA that evolves more slowly, and so it retains what we call a genetic footprint. So it can be used to look at historical movements and historical migration patterns as well. And what we did was we reviewed the mitochondrial data that had been published from across Europe to see where the Irish pine marten sit within a European context. So first of all, what we found was at the microsatellite data, so kind of our contemporary data, it showed us that the pine marten today, they have they form a single population cluster. So it means that there isn't any subpopulations or anything like that found in Ireland today. It's one big population. And that is a little bit unusual in the sense that other carnivores do actually have some level of population structure, and it's usually caused by things like fragmentation and stuff like that. And you might have expected the pine marten to have some level of fragmentation in the sense that it's more than likely expanded from kind of small population pockets, but that seems not to be the case here. It looks like it expanded from what we would have considered a single population source. The population also has what we would consider fairly average to maybe low levels of genetic diversity. They're actually, it's on a par with other Irish carnivores. And we found that there was no evidence of inbreeding. So that's a good thing. It means that there's enough animals there today to prevent them from breeding with close relatives. But I think that's where the good news ends. And what we did find was that we had some evidence of a relatively recent genetic bottleneck. Now, a bottleneck can happen in any population where closely related animals have had to breed together in order to expand from a very small population number. And there's still evidence within the pine marten population today that they have expanded from what we consider a small population. And I suppose the The thing with the population bottleneck analysis is that we can't really tell when this occurred. All we know is that it happened over the last 200 to 300 years. That's the best we can do in terms of providing a timeline on it. But it did happen. And that bottleneck, the evidence for that is still within their genetic fingerprint today. And we also had evidence that the population had a reduced population size, and that in itself, again, probably isn't overly surprising given what we know about the history of the species in the country. The other concerning feature we found was that the population has quite a low effective population size. Now, effective population size is a term that we use in conservation genetics to define how genetically viable a population will be in the future. We would consider population, an effective population size of 500 or more is important to ensure long-term population viability. And the pine marten population in Ireland had an effective population size of about 230. It's not at a level that we consider that the population is going to become, extinct in the next number of years or anything. Effective population sizes of 50 or less are of the highest, level of concern for extinction. But it's something that we need to be mindful of, and something that we want to avoid happening in the future, particularly in relation to climate change or any other kind of adaptation that the species might need to counter in the future. Then we moved on to the mitochondrial DNA. And initially what I reviewed was the study from Davison et al. In 2001 and Jordan et al. 2012. And what they reviewed or generated data for were pine marten samples across Europe, including Ireland and Britain. And they analysed just around 300 base pairs of the mitochondrial control region. And what I did then was I analysed the data and I made this a network diagram. And this network diagram places the most genetically similar haplotypes to one another. So if this haplotype P up here is the Irish haplotype, that's the haplotype that's common throughout Ireland today. And historically, we had another haplotype present in Ireland, and it was called haplotype I. And we know it was present because it was found in museum samples that were analysed in Jordan et al. In 2012. And this haplotype I had also been previously recorded in museum samples from Wales and parts of England. But it's almost, I suppose it's pretty much functionally extinct in Britain today, apart from an obscure finding of hap I in pine marten samples in a little island off the Scottish Highlands in relatively recent years. So it's safe to say that it's not a very common haplotype if it is still present in Britain. It's got these other colours here as well, and they're shared with Spain and also with Latvia. So what we can kind of conclude from that is that the haplotype P would have come from what we probably consider an Iberian origin more than likely. And if we come down here to the right-hand side, we have haplotype A, and haplotype A is the common haplotype in Britain today. It's what we consider the Scottish haplotype. And all of the animals that have been translocated within Britain have originated from the Scottish samples. So they all contain this haplotype A. And these animals, haplotype A in this analysis, showed that it was most closely related to this haplotype D here, which is found in Germany, and the haplotype C found in France. So it's kind of more of a continental type haplotype. So there was a study published in more recent times then by Reese Gonzalez et al. And they analysed a much larger section of the mitochondrial DNA, over 1,500 base pairs. And they had a much, they had a greater sample size as well. They had samples from all across the pine marten's distribution in Eurasia. They had animals from Northern Europe and into Russia, and had a good selection of animals from across the Mediterranean and Central and Eastern Europe as well. And what we found about the Irish sample is that it clustered in with this Mediterranean group. So it's this one up here. It's it was called a haplotype 20 in this analysis. And this haplotype is clustered in with the Mediterranean populations, and there are similar haplotypes found in northern Spain, Italy, and Croatia. And the Scottish or the British pine marten haplotype that's commonly found today would be this haplotype 28, and it's in the Central and Eastern European cluster of haplotypes, and it's quite distinct from what's found in Ireland today. So it's mostly kind of like, I suppose, a Central European type haplotype. So, what are the concerns about, the Irish population in terms of, the one mitochondrial haplotype that we have present? Well, we know we had a higher number of haplotypes because we found at least one in museum samples. So we know there were at least two, and we can only find one today. So it is possible that maybe there are others out there that we haven't found, but they are quite rare. When we compare the number of mitochondrial haplotypes in other Irish mustelates, otters have 9, badgers have 7, and stoats have 11. Which are all, it's a pretty high number of haplotypes, yet we only have one in the pine marten population. And when we look at the broader context within Europe, there are 69 different haplotypes found in the European population, yet we have only one of those. And the same could be said for Britain now as well, that they only have one fairly dominant haplotype there as well today. Our findings suggest that the Irish pine marten population may not have the genetic viability to sustain itself in the future. The truth of the matter is we don't know, but the insights we can get from the genetic data suggests that things aren't as good as they could be or should be. And I think it's grounds to consider the possibility of a genetic reinforcement. A genetic reinforcement would help boost the conservation status of the pine marten in Ireland to ensure that it has the genetic viability to sustain itself for future generations. We may not kind of realise the impact of low genetic diversity for, it could be a long time, but I think it's going to happen at some stage. The IUCN suggests reinforcing populations using genetically similar animals. And if we were to do that, I would suggest that we should look in northern Spain initially, as they have a high number of very diverse, but also similar haplotypes to what we have in Ireland. But obviously other areas would be worth considering as well. And I think if we were to consider a genetic reinforcement, that it wouldn't just be of benefit to the Irish population, but it would be of a benefit to the British population as well, especially as they have so many translocation projects that are happening there at the moment. And what I think in reality is happening is that a lot of animals are being relocated and translocated, but they don't necessarily have the genetic viability to sustain themselves into the future either. So I think if we did create a genetic reinforcement project, that the pool of animals that are, I suppose, generated from that kind of an effort, that they could be used for translocation projects into the future. I thought it was interesting what John was saying earlier about, maybe Ireland isn't ready for a wolf reintroduction, but surely we're ready for a genetic reinforcement of a species we already have. And perhaps it's time we gave it some consideration. Even if it's not something we do today or tomorrow, it should be something that's on the back of our minds for something that we could do, and something that would be, quite novel in a European context as well, but particularly given that Ireland and is a small island population, we're not going to kind of have some sort of a natural recovery of the species with animals bringing in new genetic diversity themselves. We're going to have to fix the problem we already created, so to speak. So that's it. Thanks very much for your time, and happy to take any questions.
That's brilliant, Denise. Thank you very much, and thank you in particular for joining us from what's clearly a sort of a darkened room and quarantine, and perhaps not too far from a box of tissues. Really appreciate it.
No,
I mean, thanks for not. We wish you were here, but thank you for not coming. But thank you for being present, and that's. I think that's real sort of mind-blowing stuff. I believe northern Spain is lovely at this time of the year. So that's personally, I'd certainly be in favour of a series of field trips, maybe over the next few years, to northern Spain, Italy, and Croatia. Was it? Sounds that sounds perfect.
I picked the nicest of them. Yeah.
Yeah, exactly. I think we need to take our time doing this research and make sure we do it right. Okay, do we have Ruth has a question from online. Yeah, thanks.
Two from online, so I'll ask the first one and let you go to the floor then. So the first one is from Emma Sheehy. What can be done to increase effective population size?
I think you actually have to introduce new blood, really. I mean, we have what we have, and we've got a limited genetic pool of animals in the sense that they've recovered from a small number of individuals. And if you do want to increase the effective population size, we do need to bring in new blood to be able to do that.
Thanks.
That's great. Thanks, Emma. I'm sure Emma knew the answer to that question, but it was a question that I was hoping to ask, so I'm glad that Emma covered it. Thanks, Emma, for coming in. We have a question here from the floor. Could you just introduce yourself, because I don't recognise all the faces.
Hi, my name's Ross. I'm from Ulster Wildlife.
Great, thanks.
Brilliant talk. Absolutely loved it. Very, very interesting. I love those beautiful colourful graphs of the haplotypes. It's wonderful. My question is the genetic viability not being possible long term. Do you have a kind of ballpark figure of when we'd visually see populations of pine martens being affected through our monitoring? If they aren't viable long term, will they have a decrease in a certain period of time in the future that will be noticeable?
You see, the thing is, we don't know. A lot of species have low genetic variability and they continue to survive. Invasive species are a good example of that. You might only have a few founding individuals, and yet they manage to create a viable population. But I think what we have is evidence that there could be a risk in the future. And I suppose we have the opportunity to maybe mitigate against that risk now in order to prevent it from happening, because otherwise I suppose we do stand a chance that it could cause problems in the long term. I don't know what those problems could be. It's just that it's something we've been able to highlight from this work, that the population isn't as genetically variable as what other Irish mustelids are, and it's certainly a lot less genetically variable than its European counterparts.
Right. Thanks,
okay.
Denise.
A final question before we go to John and Glenloy. Denise, from Francis Edwards. Could the single haplotype, different from the historical, suggest that the pine marten really pretty much died out, but for a few pairs introduced by man a few hundred years ago, perhaps to breed fur?
It's possible, yeah, we don't know. And I have had conversations with a Sardinian researcher, and he was telling me that in Sardinia, pine marten were introduced from Iberia, and they actually have the same haplotype in Sardinia as we have here. And I kind of did wonder, did people introduce them into Ireland as well? I don't know.
Okay. And thank you, Denise, and thanks for joining us despite having had COVID.
I know, yeah, I'm sorry for subjecting to that even online, but anyway, better online than in person.
I do have another question, but I will have to send it to you in the chat so that we
fine,
don't keep John too long, because I know John actually has an ecotourism event happening this afternoon. So thank you very much.
Excellent. Thank you so much, Denise. Great to see you. Look after yourself. Okay.